Okay, good morning. I hope fall break went well. So thanks for being here after that. This morning I'm going to be mainly probably entirely talking about the DNA viruses and finishing off on the DNA viruses, we hope. At the end of last lecture, we had started to talk about herpes virus. Remember, much bigger virus physically, but probably sort of more important when you come to look at what they do, a much bigger genome. And we talked about how herpes virus fuses with the plasma membrane and how, because it can fuse at physiological pH, it fuses with the plasma membrane on the way into the cell, but later on those fusion proteins can also facilitate fusion between cells. And so these are cells that cause cells to fuse and you get syncytia. And you see that for the three families that fuse with the plasma membrane. The paramyxoviruses, the herpes viruses, and HIV typically show these syncytial formation. Okay, so this is just an overview of the life cycle so that you can put it all together at the end. But I'm going to talk about each step individually, so I'm going to start at the top of here and work my way down the diagram. So here we have the herpes virus binding to the cell surface and fusing with the plasma membrane and letting its nuclear capsid enter the cytoplasm. Okay, so that's shown down here. And one thing is, yet again, the nuclear capsid takes advantage of the host cell, hitches a ride on the microtubule system, is delivered, we assume, to a nuclear pore because you can find empty nuclear capsids at nuclear pore, but you don't find them inside the nucleus. So it appears that the DNA exits the nuclear capsid and enters the nucleus as DNA, maybe with a few associated proteins. One thing here is to notice also, we said, that herpes virus is a much more elaborate virus than the ones we've talked about so far. And I mentioned there was a layer of protein, it's got some structure to it, in fact, between the nuclear capsid and the membrane. And we call that layer the tegument. And it has some proteins in it that are very important in the herpes virus life cycle. I'm not going to bore you with all of them, but I'm just going to bore you with one of them. This one here, VP16, VP means it's a virion protein, i.e. it's a structural protein. It was present in the virus particle. And that protein is released, the tegument proteins are released into the cytoplasm, and they go off and do, some of them go off and do certain jobs. This VP16 has got a nuclear localization signal on it, so it goes into the nucleus. So now you've got the DNA into the nucleus with some associated proteins, and you've got this VP16 in the nucleus. And VP16 turns out to associate with host cell transcription factors. The host cell transcription factors could recognize some of the early genes of herpes, but with very, very low efficiency. And remember, at this stage, you've got one DNA molecule in the nucleus. And you really want to get pretty efficient messages if you're going to make any decent amount of protein. One DNA is not, you want it to be copied into multiple messages and reasonably fast. So this VP16 protein associates with some host cell transcription factors and makes them work very well on the early genes of herpes virus, so that you get a lot of transcription at this early stage. And this increases the transcription from the SOSA alpha promoters. Herpes virologists appear to be very heavily influenced by their Greek alphabet. And you'll come, I suspect, when Dr. Gangemi talks to you about the herpes virus families, he's going to talk to you about the alpha herpes group and the beta herpes group and the gamma herpes group. When it comes to the genes, we have the alpha genes and the beta genes and the gamma genes. Alpha genes are the immediate early genes. In other words, those that are made immediately upon the virus entering. So they're what you called immediate early when we talked about adenovirus. Beta genes are the so-called delayed early. You need the immediate early genes in order to switch on the beta genes. So those were what we called delayed early. They are, so both the alpha and the beta are the early genes. It's just that the alpha are earlier than the beta. And then DNA synthesis will start, and you can start making the late genes. And the herpes virus people call late genes gamma genes. but it's just, unfortunately, a different nomenclature. I think there's a gradual effort to move these into the same nomenclature as the other viruses, but it's meeting a lot of resistance, as you'll see when you look at the textbooks. Okay, so VP16 goes to the nucleus. It interacts with host cell transcription factors so that they see the immediate early promoters. And then they make up messenger RNAs, which are capped, methylated, polyadenylated, because they're using the host cell RNA polymerase again. If you go to the nucleus, as I pointed out before, the RNA polymerase is there in all cells that have a nucleus. They all make messenger RNA. So if these viruses get to the nucleus, there's no particular great advantage that one can see in them not using the RNA polymerase. And as I pointed out, since it works as a complex with the capping and methylation enzymes and with the polyadenylation enzymes, it seems difficult to use any of those aspects of RNA synthesis individually because it's all tied together. So what seems to happen is the RNA polymerase functions as a factory, caps, methylates, and polyadenylates the RNA as it makes it. And if you just have an RNA that's floating about in the nucleus and hasn't been fed into the machinery at the right entrance point, it doesn't get all of those modifications. So if you want your RNA to be modified, you have to have the host RNA polymerase to make it. So if you go to the nucleus and you don't use the RNA polymerase, you have to provide all those functions as well. And it seems, as far as we're aware, none of these RNA viruses have chosen that route. It would take a lot of coding information for something that's present in all cells. When it comes to DNA synthesis, that's not present in all cells. So there's an advantage of taking your own DNA synthesis machinery in because you're not having to rely on causing this cell to divide and make its DNA synthesis machinery, or at least go into S phase. So you make these immediate early proteins, and I'm not going to go into what all of them do, but one of their important functions is to enable the beta protein promoters to be recognized. So they include proteins that modify transcription and enable those beta proteins to be recognized. So it's really a gradual change of the host transcription program from being not recognizing this to recognizing it and being very efficient on it and switching over. And in fact, in many cells, herpes viruses actually inhibit, these early proteins actually inhibit host cell RNA synthesis so that there's more available for or that's implying thought processes, but the result of that is that there's more of the reagents are available for the virus. So the immediate early RNAs go to the cytoplasm, they're translated into the alpha proteins. Those have got nuclear localization signals of some sort or other. They are shipped back into the nucleus. They then associate with these promoters for the delayed early genes or the beta genes. Those RNAs are made. They're shipped to the cytoplasm. They're translated into protein, and what happens to the beta proteins? Okay, so the beta mRNAs include genes for a lot of proteins. I'm not going to go through all of them, but one of the things is, like adenovirus, herpes virus takes in its own DNA polymerase. So it's not dependent on the host cell for DNA polymerase. But it also is not dependent on the host cell for making a lot of the other precursors that are needed. Adenovirus still needed to have the host cell make the deoxynucleotides, etc. for it, but herpes virus provides the DNA binding proteins just as adenovirus did, but it also provides a thymidine kinase which rescues nucleosides and turns them into nucleotides by phosphorylating them. So this enables the cell to salvage pre-made nucleotides from the circulation. So that's going to improve the nucleotide pool in the cell, but these are going to be ribonucleotides. So what then happens is they are converted into deoxyribonucleotides by ribonucleotide reductase, which is what you need to make your deoxyribonucleotides, which are the precursors for DNA synthesis. So herpes has got an enzyme that provides both. Now, thymidine kinase is actually a misnomer. It does phosphorylate thymidine, but it also phosphorylates a whole pile of other nucleotides. It's a very sloppy enzyme. It will take not almost any, but a huge number of different nucleoside analogues and phosphorylate them. And what that means is that if you give a nucleoside analog to the cells, the normal cells cannot phosphorylate it, so it doesn't go anywhere because it can't enter the nucleotide pool. But herpes-infected cells can phosphorylate it because they have got this very sloppy thymidine kinase. And then it goes into the nucleotide pool, and then it can inhibit the enzymes that use deoxynucleotides. So this enzyme actually provides us with a weak link that can be targeted, because if you can find a nucleoside that is not phosphorylated by the host cell but is phosphorylated by this, and that when it enters the nucleotide pool and becomes a deoxynucleotide, targets the DNA polymerase, you can just target the DNA polymerase in the herpes cells. It makes it very specific. And you'll be hearing more about this when you come to talk about antivirals for herpes viruses. But the point here is that the herpes virus has provided us again with several targets that we can try and use, and they are being used. So the current batch of anti-herpes virals target some of these. So what this does is, as I say, what you need in the early phase is to set the stage for the late phase. So what you want to do is make all of these things that you need for DNA synthesis in sufficient amount that you're ready to go and rapidly start DNA replicating. Oops. And the alpha proteins made sure that these promoters are recognized very efficiently. And so they're switching these on. And actually, the beta proteins also seem to cause a reduction in the alpha synthesis, because once you've made those alpha messages and you've turned the beta genes on, you don't need the alpha messages anymore. And so these are then turned down, presumably because you don't want to keep on making proteins you don't need at this stage. So they turn down the alpha message synthesis, so you've got to switch from alpha to beta. And now once you've made these, you can start DNA replication. So are there any questions on the early phase, the alpha and the beta phases? Okay. So DNA replication, as I said, the virus is doing it for itself, so you can expect beforehand that this is going to be rather simpler than the host cell. This is a much larger virus than adenovirus, so you may expect it will be rather more complicated than adenovirus, and both of those are the case. It codes, as I've already said, it codes for multiple proteins involved in replicating the DNA, and I'm not going to discuss all of them. But in addition to that viral DNA polymerase, it needs at least 6 other proteins to be part of that DNA replicating complex. So it's providing a lot for itself. And what the DNA replication results in, and the way these are generated is still somewhat controversial, but it results in what are called concatenars. And concatenars just mean that you've got the beginning of your genome, end of your genome, linked to beginning of your genome, end of your genome. So you've just got tandem copies of the genome in a long, long string. And the posh word for that is concatemer. And these are just long molecules containing multiple copies of the genome. And one thing that's associated with DNA replication is a huge amount of recombination. And I don't want to go into that yet, partly because it's controversial, but just to give you some idea, one thing is if you look at, I talked about the problems of replicating ends of linear chromosomes because of this business that you have to have a primer and fill in a gap. And this DNA polymerase does use a primer, and actually the herpes has to provide its own primase as well. So it turns out recombination could be a way to get around that filling in the ends of chromosomes problem. I don't want to go into the mechanism here, but it's observed in other viruses. So DNA replication is accompanied by a lot of recombination, and people are beginning to think that may play a necessary role in the life cycle of herpes. And one, possibly just a complicated result of that recombination, or possibly for some other reason that we don't understand. There's an oddity about herpes virus genomes that I need to explain to you because it explains some of the nomenclature that Dr. Gangemi is going to be using. If you look at the herpes simplex group, they have a region of the DNA which is shown in blue here, these long blue arrows, which is unique. Any sequence here is not copied again. But the bits in green at each end of this large blue arrow are repetitive. So any sequence that's in this green is in this green, and actually there are repeats within those repeats. So there's a lot of repetitive stuff here, and so the stuff in the green is represented at least twice and more than that often. And the same for the stuff in the red. So there are repeat regions, and there are regions which don't have repeats. So the regions which don't have repeats are called unique. Because there are two of them, one is called unique long, and the other is called unique short. But what we do see is even though the cell is just infected by one DNA, which is presumably in one conformation, when you look at the output virus, you can find that this can flip round. And when it flips round, presumably due to a recombination event, you've still got the same junctions here. You've still got a green block next to a red block, but actually it was the red block that was here. But since the two blocks are the same, you can't really tell. And you can also find it where this part is flipped relative to here, or you can find it where both parts are flipped relative. And so when you look at the output, whatever you put in, the output appears to be 25% approximately of each of these forms. Now, we don't know why that occurs. We don't know what its significance is. And other members of the herpes viruses, some of them may be able to flip just one region, others don't flip any regions. So we don't know what the importance of these repetitive sequences are, whether it has anything to do with the type of life cycle that these various viruses do, or what. But one of the points is if it does, if you can inhibit it, it might be a weak point, but we just don't know. But it does affect the nomenclature because people talk about genes which are UL genes and US genes just because it just tells you which part they're located in. So it's not a big deal, but it's something I need to mention. Yeah.
Kind of like transposons, bacteria?
The question was it like transposons in bacteria? Really only because there's a recombination event. I don't think you can push it any more than that. The mechanism is probably not the same. And it doesn't enable them apparently to move around the host cell genome or anything like that. So it's a recombination mechanism, but I don't think it has any more similarity to transposons than that. Would you have any comment? Okay, so that's DNA synthesis. I'm not going to go into any more details. So what you get is you started off with this linear DNA, and now you get these long concatenas of DNA in the nucleus. So you get just endless strings of this tied together. And now what you've got to do is you've got to make your virion proteins, because we've been making all the things that you need for DNA synthesis so far. So in the late phase, the virion proteins are made. Something is different about this template. We don't quite know what. But now the late genes are turned on. So the beta genes are somehow involved in turning on the late genes by mechanisms that we don't know. And the late gene messages are shipped to the cytoplasm and translated. And some of those go back into the nucleus because the DNA is going to be assembled in the nucleus, as we've seen with all these viruses so far that go to the nucleus to do things. They replicate their DNA in the nucleus because they still use some of the host cell proteins, but in this case they're using host cell proteins that are expressed in all cells, not just in dividing cells. So they're available. But again, it seems that rather than getting the DNA out, it's easier to package it in a capsid and then get the capsid out. So the DNA is going to be packaged in the nucleus, and as I pointed out before, we don't routinely ship DNA to the cytoplasm, so there's no real available machinery as far as we're aware for them to do it with. So this goes out. The late proteins are made in the cytoplasm. Some of them go into the nucleus for assembly that we're going to be talking about. But this is a membrane envelope virus, and some of these proteins are going to go via the rough endoplasmic reticulum. They're going to be made as transmembrane proteins. They're going to go from the rough endoplasmic reticulum to the Golgi, where they're going to be glycosylated. If you don't remember this, please can you review your biochemistry notes or your cell biology notes. And then from the Golgi, they're released in vesicles. The vesicles go to the membrane, fuse with the membrane, and that inserts these glycoproteins in the membrane. Okay, so let's look at, first of all, we've got to assemble the capsid. So for these late proteins, they go back into the nucleus. Actually, one of the late proteins, at least one of the late proteins, seems to switch off the beta proteins. So we have this situation. The beta switch off the alpha, the gamma switch off the beta. And so now, as I said, they're mainly structural. And one thing is, while I'm talking about this flow, what you have is the alpha protein is switched on by a protein that's in the virus particle. That's a gamma protein. So the gamma proteins, one of these gamma proteins is going to be packaged in the virus particle in the tegument. So when it gets into the cell, the gamma switches on the alpha switches on the beta switches on the gamma, and then the gamma goes into the next virus particle and switches on the alpha in the next stage. So you have this continual cascade mechanism, or passing the baton, whatever you like, where each one switches on the next one. And as I say, the beta switches off the alpha, the gamma switches down the beta. So there's this handover from each type of gene expression. Nice regulation. Yes, the protein, the question was, is the protein that's packaged that switches on the alpha, the VP16? And yes, that's right. So when this virus is packaged, one of its tegument proteins will be VP16.
That's different than the gamma protein?
Okay, sorry, I'm just VP16 is a gamma protein. And you'll find that virtually all of the VP proteins are gamma proteins because they're the proteins that are present in the virion. And by definition, the proteins in the virion are late proteins, and a lot of the late proteins are going to be the VP proteins. Most of the VP proteins will be late proteins. But there are some extra proteins that are made here because they're needed for assembly of the virus particle. You know, we said there were extra things that were needed for assembly in adenovirus. This is a much bigger, more complicated virus. And so there are a lot of proteins that are used to assemble and get it from one part of the cell to the other, but they don't make it into the final infectious virus. Question here?
It's not the one that activates the alpha gene transcription, though, right?
The question was, is VP16 the one that activates alpha gene transcription? Yes. So VP16 gets packaged in the tegument here, goes into the next cell, is released into the cytoplasm, translocates to the nucleus because it's got its own signal to get it there, and then it switches on alpha genes transcription by being a part of the transcription complex. Okay, so I just wanted to point out, I'm not going to show you the genes organization particularly. There are no blocks for early or late genes. So SV40, there was a block for early and a block for late. Adenovirus, there was no block for early genes, but all the late genes were made from a single promoter and then processed to give all the individual possible messages. In this case, we don't have any of that. In fact, there's not too much splicing goes on in herpes. I didn't put it in your notes, and don't worry about it, but just for a matter of if anybody has an interest, it seems that herpes actually keeps the host cell suppressed by suppressing splicing. So it doesn't have any spliced late RNAs because it's already cut down splicing itself. And that seems to keep our RNAs from ever making it out because they're not spliced. They can't code for what they should do. And virtually all of our RNAs need to be spliced. So this is one of the effects that the herpes has on the host cell. And as I say, I'm not going to worry you, but just for interest purposes, or to stop any more questions, whatever. Anyway. So there are no blocks for early or late genes, and apparently it's got a large enough genome that the need for alternate splicing is got round by having a large genome. So how does it get out of the cell? It assembles a scaffold structure in the nucleus, which I'm not going to go into much detail on, and then the capsid forms around that. So that's got all the capsid proteins that are going to be needed to get out of the cell. The scaffold is removed, just as you would if you were making anything with a scaffold in building or anything. The scaffold is removed. And then the DNA enters through one of the pentons. There's a specially modified penton, and it enters through one of the pentons. And it seems roughly the penton the capsid just about takes one genome full, and there's an enzyme that's a part of that specialized penton that actually clips it at the end of the genome. So it clips it into genome-sized segments. So you get a new genome in all of these. So that concatemer is sort of reeled in and then clipped at the end of each genome. And some of the tegument proteins also make it to the nucleus, and they form an initial tegument around this capsid protein. And some membrane proteins are also shipped to the nucleus, but these are not the same as the ones that are on the mature virus particle. So what happens here, and it seems complicated, but that's how the virus does it. And when you think about it, it actually makes a lot of logical sense. Somehow you've got to get this capsid across some membranes here, and then you've got to get them to the cell surface and across that membrane. And the easy way to get something across membranes, as you saw when I talked about how you get viruses into cell, is to fuse the membrane of the virus with the membrane it needs to get across. So here what happens is it buds through this membrane and acquires an envelope from the inner membrane, in the nuclear membrane. And that leaves you with an envelope virus in the space between the inner nuclear membrane and the outer nuclear membrane. And now it's got to get across the outer nuclear membrane. So this membrane now fuses with the outer nuclear membrane and releases the virus back into the cytoplasm. And as I say, it just seems the easy way to get across a membrane is either to bud through it or to fuse with it and get through the next one. So whether it depends which side you're on as to which is the way to go. But here, so you bud through it here, and then you fuse with it again, and your nuclear capsid is now in the cytoplasm. So you've got it out of the nucleus. And now it acquires a much more in the way of a tegument, and a lot of these proteins are different from the ones here. And now it buds into the Golgi. So it pushes into the Golgi and gets a new envelope from the Golgi, late Golgi. And this has already got the envelope proteins that are going to be on the final virus particle. So this virus particle with a much larger tegument, now, which acquires a lot of this, as I say, as it crosses the cytoplasm, buds in through the Golgi and gets its second membrane, or at least a new membrane. It had already lost its first membrane. It gets a new membrane, which has now got the right surface glycoproteins for it to infect another cell. This is pinched off in vesicles from the Golgi, delivered to the cell surface, just as we deliver our own plasma membrane things to the cell surface, or our own secretory things. And the vesicles fuse with the cell surface, and the contents are released, just as we release the contents of our vesicles when they're released to the surface. And that releases this envelope virus particle, and that is now ready to infect the next cell. So it's all this business about getting one membrane and losing it to get another membrane is just to make sure that it's got the right receptors on this membrane to fuse with this membrane, and the right receptors on this membrane to fuse with the next cell. Any questions? Okay. The funny thing is, now we've got this method, which used to be very controversial, and there's very good evidence for it now. You lot never seem to have much of a problem following it. The herpes virologists who got the old method in their mind just have more problems getting to grips with this. They're still fighting for the old method, and actually when you look at it, this makes much more sense. Okay. All I was really using this slide for is just to point out, we go through all this molecular biology, and sometimes it's a good idea to actually look at an infected cell, and I'm sure you'll probably see these in your pathology. But these are cells infected with cytomegalovirus. They're in the lungs of an AIDS patient, which is why there's not a lot of signs of an immune response around here, which is why the picture is actually very pretty if you want to look at a member of the herpes virus family, which cytomegalovirus is. And what you see in these is you see these large nuclear inclusions, and that was what I was just put this slide in to point out. They're so-called owl eyes, because they look somewhat like an owl looking at you. If you get two together, I guess you can see an owl. And what the stuff around the edge is actually the host cell chromatin, which is pushed out to the outside of the nucleus. So the nucleus is kind of rearranged in this. And the stuff in the middle are the excess, or the cell has not released, well, it's probably releasing virus. It's a whole pile of capsids in the nucleus. So just like we said with adenovirus and SV40, you get these inclusion bodies in the nucleus, which are all these beginning nuclear capsids, intermediate, beginning on their assembly stage, all the way up to full. Some of them have got DNA in, some don't have DNA in, but they've all got roughly the same icosahedral form, and they tend to form these semi-crystalline arrays. So these are nuclear inclusion bodies typical of DNA viruses that go to the nucleus. You see these inclusion bodies in the nucleus because they all seem to produce vast excesses of protein than what is needed, and you typically see these. You will also see some fusion, although I don't see too obvious a sign of fusion in this particular picture. So points to note, no obvious early or late gene blocks. The genes are spattered all over the place. We have a huge number of genes. That makes them much more independent than the smaller viruses that we've talked about. And since they're more independent, the big plus for us is there are far more weak links. And the more weak links, the better, as far as we're concerned, because these are targets for drugs. Currently, as far as herpes virus is going, nucleoside analogues have been the way to go because they're so easy to make and so easy to screen. But as time goes on, there are other aspects of herpes virus life cycle that we will need to target, and we're developing methods to target those as well. Okay, so are there any questions about herpes before I go into pox viruses? Okay. So, we're now going on to pox viruses, and I said before, but for those of you who weren't here, these are the true pox viruses. They cause a pile of diseases, but chickenpox is a herpes virus, so they don't cause chickenpox. So it's one of these things. Pox were originally diseases that gave rise to sort of blister-like little vesicles on the skin, and so anything that caused that kind of thing was called pox. But these are the true pox viruses. These are different members of the family. Fowlpox doesn't seem to affect us, but Orf virus is something that you can get. It just tends to cause a single or a few eruptions on the skin. It's not a huge problem, but you can get infections with Orf virus. But the ones that we are most concerned about, of course, the one that we are most concerned about is smallpox. So smallpox is a member of this family, and another member of this family is vaccinia, and that's the virus that we use to vaccinate against smallpox. It appears to have originated in an animal. It appears to be, there's a lot of controversy as to where it originally came from, but it was at least, it probably started off in cows, and so it was a pox virus of cows, which was why the milkmaids, once they'd been exposed to this, seemed to have lovely skin because they weren't exposed to they were protected against smallpox. However, that virus has been passaged in humans from one place to another, and what exactly its history now is a little bit more obscure, but it originated with the cow. So we can be infected by animal viruses, and actually Orf is one of those as well. Talking about this, a few years ago, we had an outbreak of monkeypox in this country, and monkeypox can be serious, but it's nothing like smallpox. And it was a very small outbreak, partly because CDC and all the health authorities were in there instantly. But it just shows, how easy it is to get these things happening, and why it's important that you lot keep open a good eye when you're out in the field, because surveillance and reporting of odd events is one of the things that can be critical in enabling the public health authorities to really stop these things before they become entrenched and they're beyond hope. And so here, what apparently happened was giant Gambian rats appear to be able to carry monkeypox. And I'm not very familiar with them, but they look quite cute, actually. So they were imported into this country as pets. For kids who didn't have the pocket money to buy expensive giant Gambian rats, they were buying little sweet prairie dogs. But it turns out that prairie dogs, when housed with giant Gambian rats who've got monkeypox, can pick up monkeypox as well. And then these prairie dogs were then shipped to pet suppliers all over the country. And then when the humans started saying, oh, isn't he cute, and getting bitten on the nose or something, they could pick up this monkeypox virus from these things. So we had a small outbreak of monkeypox. As it says here, the symptoms include rash, fever, chills, sores, the sort of thing that you're likely to go and see a physician about. But it's not usually fatal, fortunately. There is no vaccine for it. But once it was realized how this was spreading, it's no longer legal to import giant Gambian rats. Also, there are much more restrictions on co-housing pets with exotic pets. So the whole thing has led to a reorganization of the pet industry because it wasn't really realized that this was such a weak link. And this is just an example. As I say, these pox things typically cause these blister-like things that look very like a lot of herpes simplex lesions and herpes zoster or varicella, which is the chickenpox virus. Okay, so the pox virus, did I not have a? Well, I don't see, if you look back into the introductory lecture, there's a sort of schematic of these. But if we look at the structure of the virus, it has membrane on the outside, and it's somewhat controversial as to whether this is a single bilayer membrane or a double bilayer membrane, but it's got a membrane on the outside. It's got a core here which contains DNA and associated proteins and is thoroughly packaged in some way we don't really understand. But you can see it's got a symmetry to it. We just don't understand it. And then on either side of here, it's got lateral bodies. And we're not sure what the lateral bodies do, but we think they probably include a lot of proteins that are needed for infection when it gets into the next cell, rather like the tegument proteins of herpes. So in this case, it's not all around the core. They are packaged on either side. So in a way, if you could see this in 3 dimensions, it probably looks something rather like a red blood cell with a hollow in the middle, and the lateral bodies are filling the hollow on either side. So the DNA is in here. Okay? So we don't know how entirely it gets into the cytoplasm. That membrane may fuse with the plasma membrane, or the virus may be taken up by endocytosis, or maybe some of these viruses have the option of either way. It's been very difficult to work with, and the one we're really interested, of course, is smallpox, and working with smallpox is extremely difficult and usually illegal. You have to get very special permission to do any experiments with smallpox, and they can only be done in very special places. But it has to undergo huge review to even take it out of the freezer to do anything with it, because smallpox, as you almost certainly know, is no longer circulating in the wild, and we don't want it to get back into the wild. So somehow or other, that core gets into the cytoplasm minus the membrane. So we get early transcription, and this virus is going to be cytoplasmic. So it doesn't go to the nucleus. So as I say, straight off, you know it's going to have to code for all its DNA synthesis machinery, and it's also going to have to code for all its RNA synthesis machinery. And if it's going to modify its RNAs, it's going to have to code for its RNA modification machinery. So it's not surprising that this has got a large genome. So you have this DNA in the virus particle that gets into the cytoplasm. So what you need to do is start making some viral messages and some viral proteins if you're going to go anywhere. Since there's nothing here that will copy the DNA, you've obviously, before you can replicate it, you've got to make things to copy it. So what you need to do is make some messages. But there's nothing in the cytoplasm that's going to make the messages for you, because our message synthesis machinery is in the nucleus. So what you're going to need is to have an RNA polymerase here to copy it. And since ours is in the nucleus, this is going to have to be a viral RNA polymerase. So you need a viral polymerase. That's going to copy the DNA, recognize the immediate early promoters, copy the DNA, and make messenger RNA. But now we've got a chicken and an egg situation. Where did the viral RNA polymerase come from? Because you can't make it until you've made the message. So you have to package it in the virus particle. So the viral RNA polymerase is a part of the virion. So even though it plays no support function, it is technically a structural protein, since any protein in the virion is a structural protein. So the viral RNA polymerase is actually packaged with the DNA, and it was present in the virus particle that infected, so it's actually packaged in the virus made by the previous cell. And now it can make messages here. And you can start the early transcription. We've already pointed out this occurs in the cytoplasm, so there's no problems about shipping messages out of the nucleus. They're already in the cytoplasm. And it's done by that virally coded RNA polymerase, because we don't have an RNA polymerase that would work here. And it's packaged in the virion. And because this is virally coded, its promoters need to be recognized by its own polymerase. They don't need to be recognized by our polymerase, so they're rather different than our promoters. But it does go ahead and it makes an RNA that mimics our RNAs perfectly. So it's capped and methylated and polyadenylated. But those are all virion enzymes. Probably, we think, at some stage in the past, they captured our enzymes and have adapted them to their own purpose over time. And again, that implies that they did it on purpose, but mutations have made it more suitable for the virus, and they've been selected for. So they have features that are reminiscent of our enzymes, but they're definitely not our enzymes. And these are present in the virion. And what this means is if you take that DNA from if you take the DNA from herpes virus, for example, and you put it into the cell using lab techniques, it won't just go into the cell if you just sort of look at it or pick it up. But if you use lab techniques to get it into the cell, it's infectious, because although it uses that VP16 to increase transcription from its immediate early promoters, it's not absolutely necessary. So you'll get a few virus particles will start and the whole thing will take off, and you'll get lots of herpes viruses. So you can put herpes DNA into the cell and get new herpes viruses out, because really all the rest of the virus particle is just a delivery protection mechanism. The real thing is the DNA. But here, the DNA is what has to be replicated, but it needs those enzymes with it. So what vaccinia has to do is take the enzymes in. So if you remove the enzymes, the DNA is not copied, because we provide no enzymes to copy it. It needs its own enzymes, and they have to go in with it. So this is something that if you've got an unknown virus and you find that the DNA is not infectious, or if it is infectious, it gives you some idea of what might be going on. So it's something that is very useful in terms of putting these into various virus families. And no spliced messages are known. That's not too surprising, because the splicing machinery is in the nucleus, and this virus is already coding for a huge amount of stuff. And splicing machinery is not simple either, and it has apparently never picked up even a simple splicing mechanism. And it's got a large genome like herpes, and seems to be able to manage by just having independent genes for all the proteins it needs, rather than splicing. So those initial early gene products are made by the incoming RNA polymerase, and they play a role in the uncoating the virus further. So it seems that if you inhibit that original stuff, original early transcription in some way, the virus doesn't get uncoated, and you'll remain always making immediate early proteins. Somehow it seems that DNA is so compact or folded or arranged or something in some way or other that most of the promoters are not accessible to this virion polymerase except the immediate earlies. And one of those immediate earlies enables this DNA to sort of become decondensed and more available for the polymerase to find the other promoters, which are delayed early or early, whichever you want to call them. So once that uncoating proceeds, the other early genes are available to transcription. So the organization of this compact DNA glob, or nucleocapsid, in the virion is apparently so dense and organized that it doesn't automatically open up when it gets into the cell. It actually has to be opened up by a virus protein. So this is an unusual feature of herpes. So other early gene products, it includes the uncoating protein. Once that uncoating protein is present, then you now start making a pile of extra other early gene products, and those are involved in things like DNA replication. So it's the replication machinery. It's also the machinery for making the deoxynucleotides. It has to make its own RNA polymerase. It will need far more copies than the few it took in with it, so it makes a lot of those. It needs the modification machinery. Again, it's going to need far more copies than it took in. So the stuff for capping and polyadenylation is also in there. And a few structural proteins are made even at early times. So again, it's setting the stage for making more message, more DNA, making all your precursors, and now you go into the late phase and you start replicating your DNA. And one thing is, the replication seems to occur in a particular area of the cytoplasm. So it seems that when this initial nuclear capsid goes in and opens up, everything seems to occur, all the consequences seem to occur in a little sphere of influence around that original one. So these pictures are taken of cells that were infected with a lot of nuclear capsids so that you stood a good chance of seeing them. But what you tend to see are these so-called factories. So each infectious virus going in establishes one of these factories. And really that makes a lot of sense when you think about it, because it's spewing out all the RNAs. If they're all translated around there, the proteins are all going to be around this original place where the RNAs are made. The proteins are going to start assembling. You can see why they form in this particular in a location. So if you stain these virus-infected cells, you see these cytoplasmic inclusions, which are these manufacturing factories making new viral particles. And this picture is just to show that you don't see viral proteins in the nucleus. The viral proteins are being made in the cytoplasm. The nucleus seems not to do very much, if anything, for this virus. So here it provides all the ribosomal machinery and the genes for that and everything, but in terms of actually replicating the virus, it does very little. So these are going on in factories in the cytoplasm, which in the old days were known by light microscopy and called Guarneri bodies, because they were found by Signor Guarneri, probably a senior, not a signorina, I suspect, at the time they were discovered. So what you have are these DNA replication occurs there, and I'm not going to go into its mechanism because, as you might expect by now, it's going to be rather different from our machinery, which, of course, it's cytoplasmic for a start. It can't use any of our machinery. And also, these viruses are basically having to have some kind of simplified mechanism. And I'm not going to go into it, partly because we don't understand too much about it, but it's different from herpes, and it's different from adeno. And then once DNA replication occurs, you get the major stage, you get late transcription and translation of these late genes. And the control here is very complex. This whole virus has got very complex control. If you look at individual early genes, they don't all come up and go down at the same time. Some of them are earlier than others. Some go up and then go down. Some go up and stay up throughout the whole replication cycle, so they're still up in the late phase. Some of the genes that are mainly in the late phase started in the early phase. All I'm just telling you is it's complicated. There's no need to know more than that. But there is a crude separation into early and late. And then these viruses are assembled in the cytoplasm. It's kind of a slow process. It's thought to take about an hour to just assemble these, whereas many other viruses can do this in a few minutes. So whatever it is, however this is done, it takes a long time. And so what you find are these particles at various stages of replication. Some have got very little of this dense material in, and quite small, and then they get larger and they acquire membranes and they become more brick-shaped. So these more brick-shaped are the more mature ones. The membranes, at one stage it was thought that somehow this virus made membranes just out of the blue around it. They appear to come from cellular organelles which actually sort of surround these in some way or other. And again, how this membrane, the precise molecular details of how this membrane gets on is not clear. but all cytoplasmic, not in the nucleus. And you get them forming together because they're made around the same place, but they don't seem to form these parallel crystalline bodies because they don't have such a regular shape for packing. And these membrane-wrapped virions that you can find in the cytoplasm can be directly released by cell lysis. And they've got an attachment protein and everything, and they are infectious. Or they can bud through the cell membrane and get another membrane on them. And it turns out that this has got different glycoproteins because these are proteins that went to the plasma membrane. These are proteins that went to some internal membrane. These are different proteins, but they also can act as attachment proteins. So it seems that either type of virus is infectious. This membrane is not terribly stable and frequently comes off, so probably it's the white proteins that infect more often than the solid ones. So it's the open circle type. But both can cause the virus to be infectious. So what do I want to stress? They're very large, they're cytoplasmic, they have to do a lot of things for themselves, which if smallpox gets going again and we have to use antivirals, we will be looking at all of the things it does for itself to try and find new antivirals. And in fact, a lot of that work is going on anyway, because given the biohazard nature and the potential terrorism implications of this, the idea of having some spare antivirals on the shelf seems to be a very good and useful idea. But of course, we do have a pretty good vaccine. And it's got unusual capabilities. And as I said before, a lot of these things, whenever I say there's an exception, it's frequently the pox viruses. So if you want to sort of memorize things, you can try and memorize them. Most of them do this except for the pox. Most are nuclear except for the pox. There are other cytoplasmic DNA viruses, but in terms of human disease, these are the important ones. Okay. So I'll start on the RNA viruses tomorrow. Thank you.